So at this point, let's go over some of the things you have to be checking that you understand. At this point, you understand what biomolecules are. Also, specifically, when we're talking about macromolecules, specifically macromolecules that are also biomolecules. So, obviously, we're at this point, you should know we're talking about things like nucleic acids, and so that's DNA and RNA, and materials like proteins, and materials like carbohydrates. These all form very large macromolecules. As discussed in the video, if you wish to consider this a study guide, let's go ahead and label it study guide. Because on Friday you will have an exam on these 3 videos. Carbohydrates you need to know. You need to know cellulose, and you do need to know the type of linkage. You can go back and look at the videos. I describe it. It's in your book. All the videos are based on the stuff in your book, whether you have a new one or the old one. Glycogen and starch. Those are the 3 macromolecules in these categories. Proteins, for the exam, you do need to know the amino acids. Categories. Now, I've told you, I think a week and a half, 2 weeks ago now, that you had to know these. You have to know which amino acids are acidic, which are basic, which are hydrophilic, which are hydrophobic, and the 2 that are in between. Nucleic acids, obviously DNA and RNA, the only 2 really have to know. The difference between ribose and deoxyribose, that kind of thing. You have to know, this phosphate sugar, backbone. You have to know ATGC. You should know AUGC. You should know which are 2-ring and which are 1-ring. You should also know that the 1-ring matches with a 2-ring. Why are people leaving? 1-ring matches with 2 rings. So you get A and T, one of them is 1 ring, the other one is a 2-ring. This is simple geometry. It's literally the only way they fit together. The only way the hydrogen bonds can fit together. It's also, you should know that they're anti-parallel. So these are facts you should know for this exam and all the various things that they imply. Remember, in AP exams, they're not going to give you they're not going to give you a lot of here's a fact. Is it true or false? They're going to have you apply the facts. Is that clear? So you'll be situations where you're going to have to know that there's a 1 ring matches with a 2 ring. There might be a simple question like which one of these matches with which and why. Then you'd have to explain the geometry of it. Remember, anti-parallel means that one's upside down. That means they're parallel but upside down. What do I mean by that? I mean, if you have 5' on this end, you're going to have 3' on this end. And the opposite's going to be over here, 3' on this end, upside down, 5' on this end. Now, if you're confused about the 3 and 5', that's, remember, all about the carbon ring. It's all about the carbon ring. It's ribose. In this case, it's deoxyribose. So it's an oxygen. There's 6 carbons in total. This is this is ribose. So here you have a 1', 2', 3', 4', 5'. So it's not a 6 carbon, it's a 5 carbon ring. It's a 5 carbon sugar. This second prime has no oxygen. That's why it's called deoxyribose. The 5' has something attached to it. It has a phosphate. That means a phosphorus with 4 oxygens attached to it. This allows this oxygen now to bind to another one of these nucleotides in a base to form a chain. What do I mean by that? Let me show you. So these deoxyribose, when they start to form a chain, again, there's no oxygen on the second prime. That changes the geometry. Now, those of you that took AP chemistry, you understand that atoms, their shells of atoms repel each other. All atoms repel each other because their valence shells are negative, right? All electrons are negative. Then there's this balance between the attraction of the nucleus to the electrons and the electrons' repulsion of each other. And it's that balance and that lower energy state of the valence shell once you have sharing electrons that leads you to bonding. You can calculate actual energies. In any case, on the 3' end, you have what? Does anybody know? Out of curiosity. What's on the 3' end? That will make a connection. But what's on a 3' end? Hydroxide. There's 2 ways you know this. There's hydrogens on these as well. There's 2 ways you know that this is it, because how do we put molecules together? Let's start with the most basic understanding. Dehydration synthesis. This is a conserved trait. What that means is that life on planet Earth does this over and over again. It's one of those things that evolution, it worked, and so life just keeps using it. Like I keep telling you, when it comes to living systems on planet Earth, it's the only place we know where there are living systems. Living systems on planet Earth use the same processes over and over and over again. So dehydration synthesis means that you have to take out a water to make space for the bond. So I'm not sure if I got this right, which one of the phosphates actually forms the bond. Let's just say, for argument's sake, that this is the one that does it, okay? I don't, when you look in the book and it might be different, I don't want you to get confused. Is that fair? Okay. All right. So what's going to happen is that OH and H are going to leave as water and allowing this oxygen now to bond with the 3'. That's what's going to happen, and you're going to get this chain. Now, let's not forget that on the first prime, on number 1, prime 1, what's over here? A base. A nitrogenous base. And it's either going to be a 1 ring, and again, I don't remember the shape of the ring, so I'm just going to put a generic ring here. Don't quote me. Or it's going to be a 2 ring. Do you see how there's 2 rings? There's 1 ring here and 2 rings there. Here's just 1 ring. Do you see that? Everybody okay with that? All right, so this is a nucleotide. That is the letter. That's a nucleotide. And this is a nucleotide. These are the monomers of DNA. And it's DNA, not RNA, because there's no oxygen on the second carbon. Does that all make sense to everybody? Does anybody have any questions about that? I'm going to give you a minute to process that. And then let me know if you have questions, and I'll move on.
So the chain of the phosphorus bonding with the one nucleotide is what that's like the monomer of DNA.
Yeah, this is the monomer. That's exactly right. This is the nucleotide is the monomer of DNA. So this is the monomer, which will be what? Either A, T, G, or C.
So on the exam, you don't have to like assuming it's all this out, or do you explain how it?
You have to. I'm not sure. I haven't written the exam yet, but I would say that what I would expect you to know is that it's a 3' to 5'. Right? I would expect you to know that the bonding is from 3' to 5'. On one side, over here is what? What's the one at the end here? 5'. Let me rewrite it because the highlighting. 5'. Phosphate is attached to it. So on the 5' end, you have a phosphate sitting there. And then the 3' end has a hydroxyl. So on the 5', I expect you know that in the 5' there's a phosphate. On a 3', there's a hydroxide. And what does that give you? That gives you a direction. What that means is that now DNA has a direction. 5' to 3'. That gives you a direction, so now you can read it. Now there's a message. Without direction, there is no message. Texting in class is going to get your phone taken away and a referral to the office. And pretending like you're not texting now, it's too late. I see you. Yeah.
So you need to know that 2' has no oxygen attached to it, so it's a
I would know that. I'm not sure it's ever going to be tested on an AP exam. I have seen that you should know the difference between DNA and RNA. There are many questions on the AP exam because of that. That is one of the differences. That's right. That's right. But that is one of the differences. I feel like it's an important difference since it's rooted directly into the name. But I have not directly seen an old exam question where they ask that. And I've seen many questions. Yes, there will be extended response questions. You will have FRQs as well. Not this Friday. There'll be a short FRQ. Can you wait till somebody comes back? Somebody just left. So what we have here is what I would know for Friday is you understand what dehydration synthesis is. You understand there's a 3' end, there's a 5' end. 5' has a phosphate, 3' has hydroxide. That means there's a direction. And that also means that the opposite is happening on this side. It's going to be hard for me to draw it. So let me see if I can turn this without this thing flipping. So in order for the hydrogen bonds to work, in order for these two strands to anneal, we call it annealing, for the hydrogen bonds to stick to each other. Is that clear what I'm saying? Anneal, sticking to each other. They have to be upside down. All right, so what I like to make analogy to is two pieces of Velcro. You've all worked with Velcro, correct? Or something like Velcro. They have to be opposite, don't they? If they're the same, they don't stick. But if they're opposite, they stick.
Is that magnets or?
Magnets would be enough. It's more like magnets than Velcro, but I use Velcro because it's a string. But yeah, it's a lot like magnets because opposites attract, right?
That's why DNA has to be anti-parallel so that it can stick together.
Yeah, so they can stick together. It won't stick if they're parallel and they're not upside down. The geometry is such, right, that the hydrogen bonds cannot meet with each other. They can't attract each other. That's right. So when you look over here on the other strand, what you're going to see is, let me just copy this rather than trying to redraw it. Oh. What you're going to see on this side is the opposite. It's going to be upside down. And so if this is 3' here, would this be right? No, it wouldn't be right. It has to be upside down, like this.
That's how it works to make that hydrogen.
Because it has to be, the 3' has to be pointing down. I'm sorry, this is right here. The 3' has to be pointing down. So this hydroxide is down. Ooh, I did it wrong, didn't I?
Yeah.
My bad.
You had it right when you flipped it, because then it would be up.
Yeah, I'm just looking at this upside down, so it's hard for me. I'm sorry. So yeah, that's right. It's like so. So phosphate would have to be down in order for that to match. This is all very geometric. In order for the hydrogen bonding to occur, the phosphate has to be down in order for them to align. Also notice something else. The 2 ring matches the 1 ring. Again, these are generic drawings, so I don't want you to think this is what the nucleotides actually look like. The 2 rings match the 1 ring. When you have two rings cannot align correctly. They will not, that's why A always matches with T and G with C. Because they one is 2 ring, the other is 1 ring. And the same kind can't match the hydrogen bonds. So A can't match with A because they don't align when they're upside down.
Do we have to know specifically if A is 2 ring or?
No. I'll be honest with you. If you're interested in medicine, you're interested in moving on, and you're going to take biology in college, etc., I would know that, which is a pyrimidine, which is a purine. It's in your book. But I've never seen a question on the AP exam that asks which is a purine, which is a pyrimidine. Yeah.
So if the last, it's the 5 prime, and the pathway has to be upside down. If the 3 prime still why it should be like that upside down.
No. What's going to happen? I don't know why that there's talking when we're having your colleague asked a really great question, and I bet you that you don't even understand the question. What I would be doing is trying to understand the question that she's asking, rather than having side conversations. Or you can continue to ignore me and have your side conversation. I guess that'll work too. So, in any case, what happens is you have to have it upside down. Notice that now that they're upside down, what do you see? Now you can see the connection is possible. Now you can see 3 prime is matching with 5 prime. Do you see it? Yeah. You start with 5 prime, and then you get the 3 prime here matches this 5 prime. Then what's sticking up over here matches. And again, give me a second. Then here again, 1 ring to 2 ring. Now they're starting to line up in a way that hydrogen bonds can be formed. Is that clear to everybody? Yes. So this is why it's important that you know that it's anti-parallel. It also becomes important to understand this geometry, these arrangements, because we're going to cover DNA replication in more detail than we did in honors biology. And when we do DNA replication, you're going to have to understand, how is DNA read? How is it made? And the reason you need a primer when you're making more copies of DNA is because of this alignment issue. The DNA polymerase can only read in one direction and can only make in one direction. So you need a primer to leave an open space to allow it to bind to the proper end and start making. So understanding this basic geometry is part of the kind of a little more advanced in Unit 1, but it's still, I think, necessary that you should get that. Is everybody okay with this so far? Yeah. And the hydrogen bonds would be in between these two. If this is A and T, there would be 2 hydrogen bonds. This you need to know. It's A and T, there's 2 hydrogen bonds. If it's GC, there's 3 hydrogen bonds. So between A and T, there's only 2 hydrogen bonds. Between G and C, there's 3 hydrogen bonds. Oh, sorry. Again, I'm doing all this upside down and backwards, so let me redo that. So A and T are 2 hydrogen bonds, and G and C are 3 hydrogen bonds. That becomes important when we first discovered the bonding pair. Some of the first information that Watson and Crick used to figure out the structure of DNA was that when you have DNA or RNA that has a lot of GCs, it takes more energy to separate them. Why? If you have DNA that has a lot of G and C's, why does it take more energy to separate them? Why does it take more heat?
Because G and C are triple bonds, they're more strong.
That's right. They have more bonds. Excellent question. Don't say triple bond, though, because triple bonds suggest 3 covalent bonds, and they're not 3 covalent bonds. They're 3 hydrogen bonds. A and T hold each other less strongly. Correct. You got it. Very good. So you have 3 hooks between G and C, only 2 between A and T. That is one of the experimental results that allowed us to know how they bond, which one bonds with which. Also, the fact that you know that A, for every A, there's going to be a T. If I tell you, I've seen these questions before, so let me give you an example logic question here. If I tell you that the DNA, some random DNA, is 40% A, you should be able to tell me all the other percentages.
There's 40% of,
If there's 40% A, what else do you know?
40% T,
40% T is correct.
and then 20% G and C.
So 40% T,
And then 40,
10%.
10% is 100, right?
Yes, you would have 10% of, G,
And then is G and C because you get 100%?
yes.
that's correct.
Yeah.
So you have to be careful how they phrase it, right? And how they state their statement. But that's it. Again, this is an example of a fact that A binds with T, and they throw you a percentage, and they expect you to know how many C's are in there, even though they never mentioned C.
And it always gonna go up to 100%.
Always 100%. You can't go more. You can't have more than what you have, unless you're in a fantasy world. Are we all good?
Yeah.
All right. When you see more 101% or 102%, that's only comparing what you started with what you ended up with. If you're talking about net, if you're saying net profit, for instance, you can make a net profit of 200%. What that means is that you made 2 times the profit you made last year. That makes sense. But to say you have 200% of the DNA, how'd that happen? Like, you can't have 200% of 100%. So it depends on context. Percentages depend on context. More than 100% makes sense in certain circumstances. It does not make sense in this one. All right. We're all good with this then? And so because of all this, everything else follows. It's logic. It's simple logic. Because this is the way this is, that means the next thing has to be true, which means the next thing has to be true all the way down the line. When is this class?